or way? We're going? Okay. Well, good afternoon and welcome to the Simon's Electron Microscopy Center's Winter EM course. Today we're wrapping up our tomography section with something that we've talked about a little bit during Journal Club, and that is how do you turn something that you collect and initially process at, 3.5 nanometers and do some more processing and get to 3.5 angstroms. And that's exactly what we'll be talking about today, and that is doing Cryo-ET and sub-tomogram averaging. The speaker today, your instructor today, is Alex Noble. He's a scientist here as well as an Appian developer here, and he's been really pushing a lot of Pro-Tomo integration to our Appian pipeline, and that has been really helpful because he's uncovered a lot of interesting things as we've been taking tomograms, even if you're doing single particle. And something that's really special is that on the fly with data collection, within a half an hour of a normal data collection, you can get an initial tomogram out of that, and that's been very informative for that. So the recommendation for everyone here is take a tomogram. Even if you're doing single particle, it's always worth doing a tomogram. So Alex Noble.
Cool, thank you, Ed. Okay, great. I'm going to probably overlap with some stuff that Wei Dai talked about, but that's fine. So good reinforcement learning. All right, so why do we do cryo? Because we want to have our specimen preserved in near native or maybe even native conditions. And this is particularly useful when you're doing tomography because you can get inside cells or reconstituted environments and identify stuff at like nanometer resolution. Okay, why do we use electrons? Let's see if I can move this a little bit farther away. Okay, cool. So they're high resolution, very small wavelength. They can be focused, unlike, say, X-rays, which are more difficult to focus, and neutrons, which essentially aren't focusable. But they damage the sample, so that's a negative. Now I'm going to ask you, why would you do tomography? Like, what type of samples, if you put them in a microscope and you're like, oh, I can't do what I want to do, I need tomography. What type of samples can you imagine? I know some of you have projects with these types of samples, so in general, what do you think?
If they're big or unique, then you can't average a lot of them. You only have like one.
Yeah, uniqueness, good.
The resolution that you use smaller than mine can provide.
Yeah, that's a good way of putting it. Yeah. Other people say, like, if your resolution is too good in single particle, go down the But that's a
challenge yourself.
All right, yeah. What else? Anything else? All right.
Purification's too hard.
Purification's too hard. That's an interesting idea. So you just decide, like, let's lice this thing and just look. Maybe. All right, so yes, uniqueness. If you have heterogeneity, so if you have like a virus with some unknown stoichiometry of various spikes or ligands bound to it, then you can try single particle and try to sort stuff out, or you can just look at it in tomography and you might be able to get what you need just straight from the tomograms at that roughly nanometer or so resolution. If you have, that's what I mean by domain stoichiometry, but if you have like structural or morphological heterogeneity, so if you have a VLP that's kind of ballooning in various ways and you have a bunch of them on a grid, you can't really do single particle on it, but you can do tomography on it. Right, so you may be able to get to sub-nanometer resolution. Ed says 3.5, how do you get there? Well, first you need to have a good sample, and that's very, very difficult. All right, so why do sub-tomogram averaging? So if you have some amount of repetition in your structure, and or if you have repeating subunits with different preferred orientation to overcome various tilt range issues. So if you have, what kind of situations might you have that? If you have like an array or something, and doing single particle, you don't get other views, period. So you have to tilt your sample. You can only tilt so far because your goniometer and the entire stage only allow for usually about plus or minus 70-degree tilting. So you'll never get some of the views. So if you're completely, strictly limited by orientation, then you may just never get some of those views unless you try a non-grid technique of some sort, which is possible. Okay? So here's an example of a thing that you might want to do tomography on. If you have a tube like this, where often if you have some in vitro or ex vivo type of samples, and if they're tubes, then they might snake around, but you might find straight areas like this. And here you can pull out what is hopefully an area or different volumes that are repetitious in some way, and then go reconstruct those with that little bit of data that you have, and you can get your helical information from this small stretch. You might be able to start building in some domain structures if you have crystal structures like that. Okay, here's what I'm going to talk about today. Big overview. What limitations do we have in Cryo-ET? This also extends to Cryo-EM, but some specifics for Cryo-ET. Tilt series collection and alignment. Wei went over some of this, so I'm going to cover it a little bit differently and in different detail. Defocus estimation and CTF correction. I don't believe you've gone over this with tomography, right? Okay, so there's plenty of different ways. I'm going to explain a few of them. Sub-tomogram localization. When you have, let's say, a cellular interior, how do you go identify stuff either automatically or in some semi-automated way and with reliability? Then how do you go average all those things and maybe classify them out, try to figure out what kind of heterogeneity actually exists in your data. Then I'm going to go through a bunch of examples. And then we're going to talk about, again, processing limitations and how we can maybe overcome these. And is there software? And the answer is yes, that is trying to overcome this. This is actually a very difficult problem. I'll explain why here soon. And then some future directions and potential improvements. Okay, limitations. So we're limited by many, many things in tomography. Usually you have some type of sample that is, as somebody said, big or unique in some way. So how big can you actually go? So here are different scale bars and different EM flat images, and this is telling you how many electrons essentially can make it through your sample. So here, none, essentially, none. And then when you reach a sub-micron in thickness, then you actually get some electron transmission through your sample, and you can do something with that data. You have a lot of scattering events as your ice gets thicker, which we'll talk about in the next slide. But this just gives you a good idea of how much transmission you get through different thicknesses in sample thickness. Okay, here is a plot of, this is inelastic mean free path of an electron through ice, vitreous ice and proteins, right? And versus the energy of your electron gun. So usually if you're doing tomography, you probably want to use a 300 kV microscope with an energy filter so you can get rid of those overly scattered electrons. And you may sit down with an Arctica at 200 kV with an energy filter also, maybe for screening or maybe for actual collection. So some people did various measurements back in the day, and then our Bill Rice did a measurement here on our Cryos to see the mean free path of an electron through ice. And this is in nanometers right here. And it looks, so the mean free path of an electron through something means how far does it go on average before it hits one thing and scatters any given electron. And so here on a Cryos 300 kV, you get almost 400 nanometers thickness that you can go through. When you're down here at 100 or 120, you can't get any thicker than 200 or 250 or so nanometers before you start getting multiple scattering events being likely. All right. Second big issue with tomography is, and with, well, pretty much all cryo-EM, but you really only notice it in tomography, is sample damage. So we have to collect as many tilt images of our object as possible so that we can get as much 3D information of our object as possible. And we are limited critically by how many electrons your sample can absorb without damaging too much. And too much means what is the resolution that you want out of your thing. But at some point, you start to see these bubbles kind of nucleate at various locations inside your sample. And at that point, pretty much all of your quaternary structure is gone in those locations. So these become useless data points in your tilt series. And so for this sample, you'd be limited by at most 100 electrons per angstrom squared total across your entire tilt series. And then you have to decide how to subdivide that and what increments you want, et cetera, et cetera. And that's all based on, essentially it comes down to what resolution do you want and what kind of heterogeneity do you have in your data. Do you have something that you're trying to average, and so therefore, and is that thing that you're trying to average maybe a ribosome in various different orientations? Then who cares, it's fine, we can just collect a few images up to 45 degrees, and we can sit under that 100 electrons, no problem, because we're going to pull it out of many different tomograms and average them together. Is this a unique thing and you just need to know the structure of whatever membrane's in there or something like that, then you're pretty critically limited at this particular sample. I've seen samples that take well over 200 electrons before, actually I had a sample on last week, hit it with 200 electrons, looks pretty much exactly like it did when we started. No idea why. I've had samples where you hit it with 50 electrons, you start seeing bubbles. So it's your sample. Maybe it's the buffer, probably all the above. So this is one critical thing that you need to check whenever you do tomography. Just hit it with a bunch of electrons, see what your limit is, you have to go from there, okay? And while it's being damaged, so this is kind of like a backwards plot or something, that's strange. While it's being damaged, you're losing high resolution information first. You're starting to damage those probably carbon-carbon bonds last, but at least your side chains are getting damaged first, and then lower resolution structures getting damaged. This goes with single particle also, but with tomography it's critical because you want to collect your best data at certain locations while you're tilting. And you can take care of this both in regular single particle cryo-EM and in tomography by trying to model how much damage, how much signal to noise you have versus the accumulated dose, and then cutting off those frequencies for subsequent tilt images, essentially getting rid of the damaged parts. Okay, so this is, you can do this with various software, but it is largely heuristic, although stuff like Relion and I think EM Clarity tries to estimate it in various ways, right? So just remove damaged stuff. So all you do is you put a low-pass filter on your tilt image acquisition scheme, whatever you do. Here, collect bidirectionally 0 to 45 degrees and then 0 to negative 60 degrees for whatever reason in this particular tilt series, and we hit it with a low pass corresponding to this accumulated dose curve. And this is just a guess as per how damaged this biological specimen was. But you should be careful. One thing that surprised me in a paper by Yangzi Tan, who is also he's a grad student here in conjunction with Columbia, is he has a paper where he did this Ewald sphere correction and got to sub 2 angstrom resolution on a single particle sample, and he went through and he reconstructed his single particle sample as a virus, so it has high symmetry, so maybe that'll call into question these results, but this is interesting nonetheless. Here's the resolution on the y-axis, and this is a per frame reconstruction right here, per frame reconstruction, and I think it was a total of 20, 30 or so electrons, I'm not sure. So it was up there in the tens of electrons. He found with just individual frame reconstruction at the 20 or 30th electron, he still had 3.4 angstrom resolution. So this counters some other heuristic measurements in the past and makes me question how much data is actually lost at those high doses. But certainly at some point your specimen just starts bubbling, and you definitely have lost that data. Okay, another limitation, same limitation in all of Cryo-EM is your camera fidelity. You've seen these plots before? Or something like this, DQE? You have?
I think we really talked about DQE.
Okay, great. Let's talk about it.
I mean, I think it's been mentioned at various points, but not really done.
Yeah. So this is how well can your camera record an image, a structure, a power spectra, with respect to the frequency of the electron. And all cameras do better in the low resolution range. So 1 is like perfect recording of your of whatever image in the image plane, and 0 is pretty much failed. So over here we have, what is this, a CCD or something? Quite bad. Film over here is quite bad at high resolutions. But these new fancy cameras, we have K2 here. We don't have a K3 or a Falcon 3, but they are higher. Somewhere up there, we don't have those curves yet. Your low resolution is overrepresented in most direct electron cameras, and underrepresented, overrepresented in low resolution and underrepresented in high resolution. But this is a limitation that you have to be aware of whenever you're doing any cryo-EM processing. Okay, quick tomography. Yeah.
Do you measure those curves yourself, or is this something given to you by the cameraman?
You measure them yourself by, you put in a beam stop, and I'm
You
not
need a
sure
straight edge,
exactly what you do
ideally
here.
knife right at the sensor.
Yeah. Yeah, it's a measurement you can do. Okay, tomography, I think we've gone through a slide like this before. Collect a bunch of different angles of a single object and get a 3D structure. So what do we actually do in Cryo-EM or Cryo-ET? Have our specimen on our grid, imagine a grid there, we got our electron beam, we collect an image, we tilt the stage, we collect another image, we tilt again, we collect and collect and collect. And you tell your goniometer to tilt to a certain spot, but it's going to be off somewhere angularly, and in X and Y, and in Z. And there's going to be some error, so I'm representing those errors by little shifts in these images out over here. And so what people often do is they throw in some 5 or 10 nanometer gold beads into their sample solution before they go freeze their sample. And hopefully there's enough of those in your image, your field of view of wherever your object is, and hopefully they're not clumped together or just covering everything, such that you can use the position of those gold beads in every single image to geometrically figure out how to realign these images, because you have that error from your goniometer when you're collecting your images. The goal is to get a good quality realignment. All right? Okay. And I'll go over why you might not want to use these gold beads. I'll ask you why in a minute. Okay. You also need to be aware in tomography of the thickness of your specimen. You're usually not collecting something super thin like 10 nanometers, where you don't have to worry about this, but you're usually collecting something 100, 200, maybe 300 nanometers thick. And when you tilt your thing, your electrons are actually going through more sample. So here I've drawn it here. So this means, where do you want, so we know our sample is going to be damaged by the first 30, 50, something like that electrons. And so our highest resolution is going to be down there in the first 10 or 20 electrons. Where do you think you want to start collecting? Do you want to start at 60 degrees or 0 degrees or 30 degrees? What do you guys think? Yeah, good. And then, great. So what kind of schemes do you think, like collection schemes, are the best ways to collect tilt series? There's only a few ways that I can think of, but what do you guys think? Like, you have to tilt your stage.
There's like 3 ways.
Yeah.
One of them is like you go back and forth, like go to -3, go to 3,
Yeah.
-6 and 6. Another one is like going through -3 all the way to -60, go back to 60.
Yeah. Good, good. You can also just start at -60 and go all the way. Which do you think is the best? Or,
Going back and forth.
yeah, because what?
So then each sample has the same amount of time between electrons.
Same amount of time between what?
Between different electrons. So like if you hit -3,
Well, what does this tell you?
I'm gonna do the high angles last.
Yeah.
oh,
So that's the main
so
reason.
that's-
Yeah. So does that make sense, why? So you want to go through less ice, get the most signal that you can out of your best electrons. So you go back and forth and back and forth. That's the best collection scheme, though it can be slower and less accurate depending on what software you use. The next best is actually starting a little bit negative, so making that your first image, and then go through like this. And so you get all your low angle tilt images first. Okay. Another small limitation that Grant points out in his video is you have to take these tilt images, and then when you reconstruct, you're reconstructing onto a grid of pixels or voxels in 3D space, and you have to interpolate all these little blue points into real grid points that are these pink, purpley points. So that's a small limitation. It's not much, and you can super sample to help with this also. It's not a huge deal. Okay, play video. So here is, if I can get this. Of course not. Okay. Excellent. Okay. Let me see if I can. I hope it doesn't hurt the stream. I think I might just need to restart my PowerPoint to get this video to play. Let me try that real quick. No, it just hates this presentation mode. Okay, maybe the videos won't play for me today. That's okay. So this is just a video of a tilt series that goes like this. And then when you reconstruct that tilt series, you can go through, after you reconstruct into a 3D volume, you can look through the ice as if you were essentially walking through the 3D structure of whatever you're looking at. So this is proteosomes. This is just a single particle sample. And you can look through the XY plane, the XZ plane, the YZ plane, and the XY plane right here. And the XY plane has the best resolution of your structures because you collected the images in that direction. You don't have images of this direction, which is why you get, or the other direction, which is why you get smears like this. Here the tilt axis is vertical, so you get different looking 3D stuff in that direction than versus that direction. So in the next practical, you'll actually sit down with tomograms like this and try to understand what you're looking at. Okay, Wei went over some of the ways to collect. So here we develop and use Leginon primarily. Serialium is very, very popular. It's actually the most popular. And then there's some other tomography software out there. Okay, so tracking. When you turn your goniometer, you can't trust it. It's going to go to some slightly off angle, and the X, Y, and Z are going to be off, as I say here. And you can't use that area that you're collecting to figure out where to collect next. So you have to guess somewhat where to go next. So how do you deal with this? You can either do predictive tracking or focus position tracking. I think Wei talked about this before. Predictive is you just fully guess where you're going to go next based on where you were before. You just draw a B-spline of where you were and extrapolate to where you're going to be. And focus position is you go off to another location on your tilt axis and refocus over here and retrack over here, and then you go back to your area of interest and collect again. Okay, here is the general procedure. Put in your grid, just like single particle, collect an atlas, go zoom in somewhere, find your bacteria or whatever, and then go collect. Pretty easy, pretty standard. Okay. One thing you need to know if you're ever doing tomography collection is you need to know where your tilt axis is. And so this will allow you to put your focus on the tilt axis. Now, why would you want to put your focus on the tilt axis?
So it doesn't shift when you tilt.
Good. Good. Yeah. So eccentricity, that's correct. So if you focused, like, it's hard to do this one. If you focused over here, then you would get really good, what we call eccentricity, where my hand isn't going up and down in Z while I turn. So that's exactly why we focus on the tilt axis, is so your sample doesn't move in X or Y or Z off from where you expect it to when you turn your sample. Okay. So you have a bunch of tilt images. How do you align them? So you can use a software called Tomo to align using fiducials. There's also AUTOM for fiducials. These both work great. And I think EMAN2 can use maybe fiducials, but it does some object tracking. Interesting protocol that I don't think is released. eTomo and iMod also have patch tracking, which is a correlation-based nearest neighbor geometric tracking. Remember, with fiducials, you're just trying to locate the things in 2D and then extrapolate or go backwards into 3D and figure out where those gold beads are. And you can do the same thing in a more remedial way with little patches of your images and try to track the patches of your images. It doesn't work so great. There is Tomo, which is completely fiducials alignment, and I'll explain exactly how that works in a minute. There's another patch tracking with GPU acceleration called Alignator, and everything must refine for the shifts, for the rotation, or actually in plane rotation. So you have this rotation and you have this rotation on a per image basis. For magnification changes, and why might magnification change with different tilt images?
Have like the edge and like it closer.
Yes, that's correct. So, so what he's saying essentially is, if you take your sample and you just actually move it up in the microscope, you'll get different magnification. If you move it down, you'll get a different magnification. I'm not sure if it's bigger or smaller. I got to think about that. But if you tilt it, then you actually have a gradient essentially of magnification over your tilt image. And so you have to correct for that. You also have to correct for your tilt axis location. So if you didn't get perfect eccentricity, if your tilt axis is actually over here, then you have to do some correction from there. Or that's off axis. For tilt axis, we call it tilt azimuth. It's saying where exactly actually is your tilt azimuth, and this can be different for different angles even. And I mentioned already tilt angles. So there's a lot you need to track and refine in order to reconstruct a tilt series. So here, this is iMod. If you have these gold beads in your sample, then you need to go in a semi-manual manner and identify them in all of your images and make sure they correspond to each other so that you can track them back in 3D and reconstruct your object. But there's some issues, I'd say many, with gold fiducials. Gold fiducials might move relative to your sample. So here, you might be able to see in this 2-electron image, this is a typical contrast of a 2-electron image. Here is whatever this sample is, some type of membrane, extruded membrane, and we have a bunch of gold beads right here. And this is without frame alignment, and this is with frame alignment. It may be true that the gold beads are moving differently than your sample when getting hit with electrons. And so this is mainly because your gold beads are absorbing electrons, that's why they're black. Electrons don't actually make it go through, or don't actually make it through. Your sample primarily, well, it's primarily transparent to electrons, and then secondarily it scatters electrons elastically, or rather inelastically, and then tertiarily elastically. And so you might get different movement of your objects of interest versus your gold beads. And that movement might be anisotropic, meaning that it's not just one direction. Here we have an example of, I forget if that's pre or post frame alignment, beam-induced motion of your gold beads and maybe of your sample. And another issue, you might have aggregation in your gold beads. You might want to collect here, but you don't have any gold beads to align with. They're all sitting over here. Or you might want to collect in an area where there's too many gold beads. These are, yeah.
So historically, they use these gold beads for tomography
People
forever
use it all the
before
Yeah.
recently. Have they reprocessed data with these gold beads in it? Can you track on just the sample itself, excluding the gold beads?
How do you, so subtracting gold beads from your images is what you're saying?
Or just, is there any like smart software that can just ignore them so that you could reprocess like historical data basically?
It's hard to ignore something that's in your power spectrum, because just cropping it out of the real space presents hard edges, and it's not I don't know of anybody who's done that, because there's really no motivation to do it. But it's not simple. Yeah.
Okay.
There's entire papers on trying to subtract gold beads and other junk in tomograms. But yeah, I don't have a good answer other than that. Yeah.
Also, a lot of people just annotate them. So if you digitize them, you don't care to trace around the gold bead.
Yeah.
Because you're not looking at the raw tomogram, you're looking at a digitized version of it.
Sure. Okay.
Yeah.
I assume people are like experimenting with other types of material beads.
Material. So the whole
Don't
point
need
of a
to
gold
now.
bead
Yeah.
is, well, I mean, the whole point of a gold bead, Experimenting for what reason?
To see if other types of materials have like lower aggregation or lower beam-induced movement.
I don't know of anybody experimenting in this way.
Most people just use gold.
I mean, gold is. Yeah.
Unless you want to do clem, that's actually a use case where they try to do some light microscopy and hopefully it's also EM sensitive or electron dense.
Yeah.
To a varying degree.
Not many examples.
No, it's not very easy.
Yeah, you need something that's electron dense and biologically inert, hopefully. And there's not much.
Yeah.
There's not much.
No. Okay, here's another issue with gold beads. Is let's say this is your object of interest, whatever, and here in this picture I have a few. These are viruses. These gold beads are now messing up these viruses in 3D. They're screwing up both the real space, which is what you see here, with these rings around them, and in Fourier space, it's messing them up. So you probably wouldn't use these viruses if you're going to go do post-processing. And even if your gold beads are far away from your object of interest, they're still in the projection of some of your images. So that shows up in your Fourier space. And sometimes actually in your real space, if you don't have a perfect alignment of these images, then you'll see streaks because like this image is slightly misaligned and it will be streaked across your 3D area. Okay. Now there's some methods for patch tracking where you try to identify local areas that hopefully don't move and are present in every single tilt image, and then use those local areas as 3D beads essentially. But it's completely sample dependent. It seems like you need features that are visible that actually confirm that it's actually working, and it's overall not super reliable, but it might work for some cases. Okay, then per Tomo alignment. Let me explain how this works. You collect a tilt series like so. So per Tomo is completely without fiducials. It's something I've integrated in Appian here. And what it does is it takes the first 3 images and it just does nearest neighbor correlation. So now you have 3 images at slightly different tilts. And then you take these and you, what you call weighted back project them into 3D space. So this is like a reconstruction just with 3 images. So imagine if you had 2 electrons per image here. So now you have 6 electrons effectively back here. And then you take a sub-volume of this 3D space, and then you reproject that sub-volume in the direction of the next tilt image, and then you align your real tilt image to that reprojection right there. And then you add your newly aligned tilt image back here, so now you have effectively 8 electrons a signal. And then you keep doing this back and forth, reproject, align, back project, all the way up until you align all your tilt images, and then you do this entire procedure iteratively. And after every iteration, Tomo tries to reorient your objects in that 3D box to maximize the signal, and it tries to refine the tilt azimuth wherever your real tilt axis is. So it keeps track of all these geometric parameters except for the tilt angle. It doesn't refine that tilt angle. So you iteratively do all of this, and if you have enough signal back here, then you can align almost anything with this method. Okay, so these videos might not play. We might have a lot of missing videos today, for whatever reason. Because they play if I'm not in presentation mode.
But you might be able to do it.
If you want to just pop out.
Yeah.
Up to you.
Whatever you do.
Yeah, I'm just gonna pop
You
out.
can still see it on the YouTube channel. It's fine.
Yeah. Oh wait, am I able just to?
Yeah, you can have it like this. You can still see it.
Is that
No,
actually?
now we're out because you have extended desktop. It's no longer.
Oh. Wait, but I see it in the video player.
Yeah, I can see it, Ed.
That's fine. That works.
Yeah, that's all we want.
Oh no, wait. That's actually my screen. Oh, I
It's your
see.
screen. You don't see. There's a 30-second delay, so I saw you move it off. onto that
Oh,
second
so you
screen.
want it?
You
Oh,
would have
okay.
to mirror displays.
Here, I will mirror my display. Whatever. Good for YouTube here. Okay, good.
Well, I know because the ingestion is here, so if it's not here, then it's not on the YouTube channel.
Yeah. Okay, good. So,
Good. We're good.
oh, and now it doesn't play.
That's
No,
the oddest.
it's just messed up too.
That's just how we're gonna have. I can't show tomograms today. Yeah.
Sure, it's not important.
Really?
Program talk.
Yeah, no,
Yeah,
it's
why
not
would you do
important
this?
at all. All right, great, cool, very cool. Does it show in this view? Oh.
Yay!
Okay, but now it's showing the sub view. Oh well.
Oh, it doesn't
YouTube
matter.
gets the duplicate view.
Gets the duplicate.
Too bad. This is the only way it works. Okay.
Yeah.
So why is this fancy method useful compared to like patch tracking? So if you don't have any gold beads, then great. You can still align your tilt series. If you do a nearest neighbor kind of patch tracking alignment, where you just take image 1 and 2, align them 2 and 3, 3, 4, etc., and just nearest neighbor align everything, then you're working in correlation space with this much information. You see this little dot that is moving around right here? That is the real correlation of those two images. These streaks are caused by various hard edges in the actual signal in each tilt image. And so you're telling a computer, try to find this dot that is faint and floating around, and it's going to have a very difficult time, and it's not going to be able to refine different geometric parameters of your stage and every sample. With Tomo alignment, you get, after refinement, you get a very nice peak right there. All the computer's trying to do is find essentially the brightest point or the center of that peak, and by doing so, it can align. This is a sample of some liposomes with some reconstituted cell adhesion molecules in them. And so like between 50 and 150 or 200 nanometers thick on the bottom right. All right, so that alignment method allows you, enables some projects and can significantly expedite some other projects because you don't have to collect by gold beads. It does have some issues. Potential issues is if you have a really streaky peak like this, then often you get a systematic offset where it's trying to find the brightest pixel, and this is only at higher tilt angles, and that offset will be like to the right in this case, and that will produce, instead of spherical stuff, kind of elongated stuff. So you can solve this just by trying to find the streaky peak itself and then finding the center of that. So there's little issues that you might run into and should be aware of. Okay, defocus estimation. How much of this have you got? Yeah.
How well does Tomo do compared to fiducial alignment?
This is always asked. And,
I haven't heard it asked yet. So.
Yes, yeah, good call. So I don't think anybody's done a direct test of like a real sample.
Right.
I think I've heard of somebody doing a synthetic sample. Somebody has done a real test of dual tilt series alignment, where if you collect on one axis your sample, and then you physically rotate your sample 90 degrees, and so now your tilt axis is, so you collect here first, and then you rotate it entirely like that, and then you get these angles right here. It gets you better 3D reconstruction. And somebody has done Tomo versus fiducials there. And Tomo does better, but it does better because of the order in which it aligns things. What the fiducial alignment dual tilt axis does is it aligns this tilt series, and then it aligns this tilt series, and then it combines those two aligned tilt series. What Tomo does is it does that back and forth algorithm, except for, I think, all 4 directions. So it may be an unfair comparison, but it does do better in practice in dual tilt. With single tilt, I don't know, but I'm going to show you later why I don't think it matters for most samples. As long as you have something to pull out and subvolume align and average, then you can do post-processing that makes it irrelevant.
Okay.
Yeah.
So if you wanted to do, you had a sample that you wanted to do some tomogram averaging with, you get like
The best
3
whatever.
angstrom
Yeah.
resolution, whatever. Tomo is fine, it aligns.
As long as you can
tomogram.
get stuff aligned such that you can pick
Right.
your particles,
Yeah.
then you should be able to go to, I'll talk about quickly, EM Clarity or Warp or EMAN2 to do a sub-tilt series alignment.
Okay.
Then after you do sub-tilt series alignment, it doesn't matter where you start your sub-tilt series
Right,
alignment,
yeah.
you're doing those little refinements and you're going to get effectively to the same
The
place.
same thing.
Yeah.
Okay.
That's what I argue whenever anybody asks this.
I really ask, again, it's a roundabout way of asking, why the hell would anybody use fiducials at all if Protomo exists? Because I still see people using fiducials all the time.
It's faster. So at least right now, it's faster.
Okay.
Protomo currently
Computationally,
is,
it's faster.
yeah, Protomo is currently CPU, though there's people working on GPU.
Right.
And fiducials, you can align a tilt series and almost completely automate it, one button click in 5-10 minutes.
Protomo takes a little bit of work.
Protomo takes some work depending on your sample.
Right.
Yeah. Youngzy has the hardest type of sample, and it does take extra work. But it's possible. Okay, right, defocus estimation. So have you guys talked about defocus estimation? Yes. And so what are you trying to do when you estimate defocus? Like, what are you actually estimating? Because this matters in tomography. Because this thing that you're estimating is changing for everything all the time. So what are you actually trying to estimate?
Contrast transfer.
Right, and what is that actually telling you about your sample?
Which spatial frequencies are present?
True, but physically in X, Y, Z, what is it actually telling you?
Which information you have?
Sure, sure. What happens when you go into focus? Do you have any issues with your information transfer in terms of zeros?
Your contrast.
Think about what we did when we found eccentric height of the sample during the microscope practical.
Okay, maybe I'm asking a slightly convoluted question.
No, I think it's a good question.
Yeah.
I think it is.
The answer is, it has a one letter answer. What are you trying to figure out about your sample? Boom. Z height. Yeah. Is that where it is in the column of microscope?
So finding defocus is not really that fundamentally different from finding the eccentric height when we do that in the microscope. And we're talking about tomography, which is basically what we use to find eccentric height, which was rotate it back and forth and then find that spot where it doesn't swing and it stops moving. So eccentric height is basically Z height, and defocus is basically the microscope focusing at a different position in Z.
I haven't noticed Alex put it in the slides.
What?
It does say
It does
in the
say
slide,
that.
find the height of your
Folded. Okay, great. So that's all you're trying. So whenever you're sitting down and you're like, what am I trying to do with defocus? You're just trying to find the height of your objects, and you need to know the height so you can correct, like you said, for those flip overs in your frequency space. Okay, that's all. Good. So in tomography, so in regular single particle, you just collect and maybe you have a tilted sample, and so you estimate the defocus and that tilt. You're done. Super easy in comparison. In tomography, I've listed, and maybe I should have done an animation where I don't show them all at once, so you don't have a giant list right here. But here's the giant list. I've listed a bunch of ways to actually estimate defocus that people have come up with. One way, from worst to best, roughly, is per image. So just take every single image, look at the power spectra like this, and estimate the defocus. Find the zeros, use some software like GCTF, estimate the defocus. Now, why would that be the worst? It would be
lower test per image, so it's harder to see that.
That's one reason, yeah. There's at least one more good reason. Hmm?
The gradient.
Yeah, gradient. So, since, like I said, you're just trying to find the height of your objects, a tilted sample has multiple different heights. And so what effectively happens is you wash out all this higher resolution information, and you might only end up with one tone ring to estimate anything from at your higher tilts. Okay? Second way, if you go through, do no CTF correction or defocus or anything estimation, you pull out all your objects of interest on a per tomogram basis, reconstruct those and then look at the FSC curve. Have you guys done FSC curves yet? I forget. No. Well, it's a way to effectively figure out the, in this scenario, the frequencies in common with all of your objects that you're averaging together. Okay, and so what effectively happens is you get a dip at the first zero because it says this frequency is anti-in-common. And then from there, you can say, okay, I know where my first zero is, and correct it. So it's actually a decent way that can get you sub-nanometer resolution. Okay, here's a way that, so assuming that you collect with good eccentricity, so you're not collecting like this, then you can take all of your images and overcome that limited dose per image. So like if you have 2 electrons per image, that's not great, you're going to get a crappy estimation. But if you can combine all of your images, by assuming that you have eccentricity, what you do is you take little tiles, little square tiles, and you cut out little bits of every single image at every single tilt. And then you know geometrically, you know your field of view, like a micron or something like that, like micron across by a micron height. You know where your tilt axis is. And so therefore, at like 45-degree tilt angle, you know where this little tile is in Z. And so then you can bring every tile, like a tile down here, a tile over here, a tile in the middle, you can, with those known Z's, and assuming eccentricity, you can bring them all back to where the center of the 0-degree image is in height. And then you can combine all those power spectra together. And so therefore you'll have a power spectra of all your tilt images. You can actually, usually with almost any decent sample, I can estimate past half Nyquist, which is pretty good for just a single tilt series. All right. You can collect, this is an arduous and time-consuming process, but it may be useful for certain samples. If this is your tilt axis and your area of interest is here, then you collect here, you turn your goniometer, and then you go focus over here, and then you focus over here, and then you find the average, and then you use this for tracking, and you use these 2 high-dose focus images for post-processing, and you take the average, and you assume that the average is the actual height of your object. So this is something that was done back then and in the Rely on Single Particle Tomography paper. All right, you can do per particle, and this is difficult to do, where you pull all your individual particles out of every single tilt image and do a per particle estimation of the defocus, and account for the 3D location in the actual volume. So your objects are going to be in different locations and they're going to be tilted, and so you can try to account for all of this. There's software that attempts to do this, luckily. And what's this extra thing? Oh, right. Taking it, so you can do this and try to take into account overlapping objects. So in a cell, you'll probably run into a ton of ribosomes that are overlapping in different projections, where you have like 3 ribosomes right here, and you're trying to estimate the defocus of this ribosome up here, and this ribosome here and here. And you can attempt to do some type of signal subtraction to get rid of these 2 ribosomes, so you can estimate this ribosome right here. That may be possible. I don't know of any software that does this, but this is just me imagining. This is the only one that I don't think is implemented anywhere. Okay, CTF correction methods. Good. So again, from worst to best, if you did per image estimation, you can do per image correction. It's not going to be great. You can do a strip-based correction. So if you do the tiling, where you assume eccentricity and you tile, and you get the defocus at 0 degrees at the center of your stage, then you can then extrapolate back. You say, this is my defocus, and then you can extrapolate correction strips, because you need to correct for every different height of every single tilt image. And so you do it by strips, where each strip is a slightly different defocus. This is implemented in iMod and Tomo CTF. Right, and so your error will be how much non-eccentricity you have, and you're actually not taking into account the thickness of the ice when you're correcting in this way. Okay, you could do a 3D CTF model, which is something that is built into Rely on if you give it the correct values, essentially, your defocus. It will do, it will assume whatever defocus you give it on a per tilt image basis is correct, and it will take into account the locations of your object in 3D. All right, you can do a per particle, essentially a local tiling CTF estimation and correction. EMAN 2 has something like this implemented. You can CTF correct during reconstruction, so you can make a CTF corrected reconstruction. This is done in a couple of different ways. The most successful is Nova CTF, where it takes the tilt series, it makes multiple different copies of the tilt series with different defocus corrections, and then it decides based on the location of your objects in 3D which different defoci tilt image to use in that part of the reconstruction. It effectively makes a 3D reconstruction that is CTF corrected, taking into account all the tilt issues and your 3D locations of your objects. So it's a really innovative way to correct for CTF, and it's resulted in some of the highest resolution structures, about 3.2 angstrom, I believe. And you can also do just a per particle, like actual per particle. I think this is more of a tiling. This is, I think it's takes just the per particle tilt image at every tilt angle into account when estimating and correcting for CTF. And that's gone to, I think, 3.1, I want to say. Okay, so that was defocus estimation and CTF correction. Are there questions there? This is, so the entire field, as you might have noticed already, is in constant development, and it's very ad hoc, and yeah, this is how it is now. If you have better ideas, you can write up your own software and add to the pile. Okay, so you pull your objects out, you know, your ribosomes or whatever your objects of interest are. In this case, like isovirus and pulling out some spikes. This should be cited, yeah, Briggs. And they're all in various orientations, and your goal, just like in single particle, except here you have 3D information, is to align them all in the same orientation. So you go through the same steps where you start with some initial reference, maybe a blob or maybe something directly from your data that you can reliably align to. You align to that, and then you iteratively keep refining that alignment and then reference, until you reach some convergence. And then actually what's nice about tomography is then you can take your average, or maybe you did classification, you got a bunch of classes, and you can map them back to the 3D space of your tomogram. So you can make beautiful movies of real averages back into your tomogram, which might be a cellular environment, and it might tell you relational information about your biology. Now here you have to take into account the missing wedge. So in this example here, your missing wedge might be, so you guys have heard the term missing wedge, you know what I'm talking about? Okay, good. It might be in this direction roughly, and so every particle will have it by default in this direction, but then when you align them all, they're all in different directions. And missing wedge is telling you that I know I have information in here, but I don't have any information up here. And so you just have to make sure, and all the software out there does this as long as you use it properly, that you tell it, use this information, don't use this information, which means that you're going to align to this in common information, and you're going to ignore the rest. Okay, and actually, regular single particle cryo-EM is pretty much tomography where you have a giant missing wedge. So tomography, we're actually, yeah, everything is tomography. Don't listen to the single particle. Okay, good. Here's a visual, the visual effect of what happens when you have a missing wedge on a 2D image. Remember, we're dealing with 3D structures, so it's a bit different. You see these streaks that are actually in the direction of your missing wedge edge, and you can see them here, see them right about here, and then if you combine them together, you kind of get a convolution of the two.
Is the sub-tomogram averaged on many pictures? Like for B?
Sorry, so you say that again?
Say like for B, you took many pictures of randomly oriented pictures with missing wedge, and then you average them and you get that.
Yeah, if you take B and C and you average them, you get that. That's what's happening.
No, I'm saying like, so you have multiple pictures of B,
Okay.
right? Of
Yeah.
Picture of Einstein. You have
Okay.
all of them have different orientations.
Oh, if Einstein has different orientations, yeah. So if these are ribosomes, like
Yeah,
floating
say
around
these are
inside of
ribosomes, and then you average them all together. That's what B would look like. That's what you had.
No. If you just had one Einstein in one orientation, and you apply this missing wedge, then that's what you
If you
get.
go to 46,
This is just
like
one
take
Einstein.
out that chunk of information.
Yeah,
That's
so this
what it
is
looks like.
one Einstein, no missing information. One Einstein, this missing information. Another Einstein with or the same Einstein, different missing information. B plus C equal D. Yeah. So that's all. Okay, there's a bunch of. We're not done with software. We have a million. I couldn't list them all even. So you have your tomograms. You want to pull things out. What should you use? There's a few different ways you can identify and pick stuff out. I prefer Dynamo for a lot of projects because it allows you to pick on like helical stuff. It allows you to pick on membranes. It allows you to pick down tubes and on the surfaces of vesicles. It's very extensive in its picking capabilities, and it runs on GPU in the sub-volume alignment part, actual multi-GPU if you have it. Relion doesn't have picking abilities as far as I know, but it has this 3D CTF model, and it uses the normal Bayesian approach that is used in the single particle Relion, so it can be quite robust in classification. EMAN2 has a sub-tilt series type of refinement and defocus estimation and correction by tiling. That seems to be, at least Muon can use it quite well. Muon's the developer of the tomography part of EMAN2. EM Clarity, which is developed by Ben Himes, who's currently in Nico's lab at Genelia, soon to be UMass, has this quite successful and actually released about a year ago, and I've heard quite very good things about it, and I think you should give it a shot. You need a lot of GPU RAM, but if you can do that, then you can start refining, essentially do a single particle constrained refinement called sub-tilt series refinement, and I think probably better defocus estimation than the others. Tigris is something that is from the DeCaster group at University of Southwest in Texas somewhere, and it's intended for you to take a high-dose 0-degree image. It's like 20 electrons, and then collect your 2 electron, 2 electron, 2 electron. Use this tilt information to get the 3D locations and maybe initial models of your object, and then revert back to that high dose 20 electron or whatever 0-degree tilt image. There's some older but highly used software, Python, Pete, I haven't touched these very much, Jason Tomo, and some others. And there's also Warp, but that's like never going to be released as far as I know. Okay, so here is how Relion does it. I explained this, should just go into the slide instead of using my arm, but whatever. So for every single tilt image, you take a high defocus image right there, and use it for tracking and use it for defocus estimation, assuming that it's just the average. And then you build up in 3D. So each of these slices here, this is essentially your missing wedge, but you actually have a bunch of little missing wedges because you're going like 2 degrees, 4 degrees, 6 degrees, etc., etc., etc. So this takes into account all those little missing wedges, and it takes into account the CTF, which are these ripples that you see here, which they got from the averaging these two images on a per tilt image basis. And the particle height is taken into account too, so you have to rely on the XYZ location of every particle in addition to the defocus. Okay? And as I mentioned in the beginning, you should be correcting for the dose, the damage that you're of electrons are doing on your sample. And so remember, in frequency space, you have your high frequencies on the edge, your low frequencies in the center. And so what's effectively happening here is the first image that was collected was a 0-degree image, and you keep all of those frequencies. And then as you collect more and more and more, you start to cut off those frequencies. And then they went back to 0 degrees and collected the other side. And you just kept cutting off these frequencies. And I think rely on, well, it will use an imposed dose curve like this. But I think it might have a Bayesian estimation of, I know it does a single particle, but I'm not sure if it's been translated to tomography, where it tries to figure out heuristically in some way, algorithmically, how much dose or how much signal actually exists on a per tilt image basis of every particle. Here's some results they show in paper. With no CTF correction, 18 angstroms is some highly symmetric virus. With, let's see, unweighting, so this is without the dose weighting, you get 10 angstrom. With that dose weighting, you get an extra angstrom out of it. With the IMOD or Tomo CTF strip-based CTF correction, that doesn't take into account the thickness of the ice, where your objects are in X, Y, and Z. You reach 11 angstroms, and if you do this 3D CTF, then you reach about 9 angstroms. And then they also did a gold versus carbon comparison where they showed an increase of about an angstrom. Okay, great. What else was I showing here? Oh, they did a comparison of a 6-electron 0-degree image essentially, where they just took this into regular single particle to get to 6.5 angstrom for whatever reason. Okay, in EMAN2, they break up every single tilt image, or at least this was from about a couple years ago or so. Break up every single tilt image into little tiles, try to identify which tiles you maybe should or shouldn't use, and then estimate and correct based on those tiles. And they reach some good fraction of Nyquist here, and here on a per tomogram basis they show in this paper. So it's quite a functional method. Okay, great. Annotation, and so imagine you have a cell and you want to annotate it. You want to get one of those nice pictures where you have all the membranes, all the whatever nucleosomes and whatever you have in your cell, a bunch of tubes and stuff. How do you do it? So Dynamo, you can go through and you can do it in a semi-manual manner, where you can just draw down the center of tubes, or draw just little layer lines on the surface, and then we'll make that full surface with all the curvature to it. You can pick out vesicles and everything. I like Dynamo a lot. EMAN2 has quite a functional, shallow deep learning neural network where you draw a few of your objects of, I'll actually show in the next few slides, your objects of interest, and then you tell it to learn what those things look like, and then it'll go pick out through your entire tomogram. And you can do it in a competitive manner, I believe, so you can pick out different things that actually look somewhat similar reliably. Amira, which is nice and expensive and is completely manual. I think Thermo sells it, of course. And so we have a copy of Amira here. It's on our back left workstation in the control room, and you can actually sit down and draw every single slice of your tomogram. And when you have a tomogram, you usually have hundreds or thousands of slices. So we've had people there several times, months at a time, drawing their tomograms. You can use some, I would say, slightly rudimentary methods of just using a threshold-based segmentation, like in Chimera, with a piece of software called Segger. You can also just use template matching and say, here's my thing, go try to find them, and hopefully it finds them. It might work. Go ahead and try. So in Dynamo, which is so every about twice a year or so, I give a tomography workshop where I go through Appian per Tomo and actually collection a little bit, and then Dynamo tutorial. It's all hands-on. So we do this a couple times a year, hands-on. You can just identify the center of your, in this case, squiggly tube, and then you can say, okay, I have a radius, and here's my helical parameters. You can actually, there's some scripts in there where you can estimate the helical parameters, which is wonderful. And then you can just go pick on, you can either pick down the center like here, or you can draw out the helical boundaries and pick each of these little dots as if it was its own little subparticle. Okay, and you can pick membranes like I was mentioning before. So this is like a full membrane right here, and all you have to do is draw little layer lines, and then it'll take those layer lines, it'll interpolate equally spaced points, and then it'll draw triangles between all those, and you can refine those triangles, and then you can tell it actually to pick on that membrane surface, and it'll actually have those picks all pre-oriented to be normal to that surface, which is wonderful and exactly what you need in most scenarios. It's very versatile and functional software. You can pick vesicles, so you can just say this is the center of my thing, here's the radius. Even if they have all different radii, you can do this pretty quickly, and you can pick in usually an overpick manner, and then do some sub-volume alignment and classification and de-overpick yourself. Here is EMAN2's shallow deep learning. What you do, essentially, if you want to pick out these tubes right here, you give it some training data, you say these are my tubes, and then you identify some regions where these are not your tubes, and then you say, okay, learn on those two things, and it will go figure it out. And now this is the neural network output right here in this 2D slice and in the 3D full tomogram, and as individual learning networks, essentially. And you put them all together and you get that beautiful tomogram right there. So this is not like hypothetical or anything. You can sit down and do this pretty decently for most data. So it's great for visualization, and I think you're able to use these kind of coordinates that the neural network outputs, maybe do some extraction based on those. And they do have geometric parameters, so you should be able to. Alright, so all it's showing here is, alright, take this, sit down and just draw those two lines. That's part of your input, and then you give it a few of these lines as input and one negative or multiple negatives for output. And that's what it is for after it trained, that's what it decided to pick. Then you pretty much click a button. I mean, Micah actually did this. I think it's pretty much that easy. Do that, and do it for all your other objects. So that's been a good addition just a couple years ago to the field. You can do template matching. This has been around forever. You have a known low-res, let's say, structure of your thing, and you just say, go match this in the tomogram, and you can repeat them together. So if you have multiple different things that look slightly different, then you can pull them out. This is nice because it'll give you orientational information of your objects. And so if it does identify it well, then you can pull them out with them pre-oriented already. Okay, here we're going to jump into examples. So any questions on how to, or anything we did up to this point? Cool, good. All right, here's an example from the literature. I'm going to go through several examples from literature and a few from in-house. So here we have some, what do you call it, heterogeneity in morphology, essentially the shape and the size of these, what are these, probably viruses, oh yeah, COP1 vesicles, great. And they're interested in the structure of the outer coat here. So I believe they did some overpicking where you don't know the actual periodicity of these objects, or you claim not to know, so as to do a de novo extraction and alignment. And then you go through sub-volume alignment after you pick them all out, and then classification, and then you draw a distance cutoff between your overpicked objects in order to get unique picks, and then you can go model these back onto the individual vesicles in this case, and you can put them back in the tomogram if you wish to. Alright, here is this is from Briggs here. Florian Schur and John Briggs' lab. He's running a lab now in Austria, I believe, where you overpick on these tubes, and then you align all those objects on the tube, whatever these are, gag proteins, yeah. to each other until you get a nice, what you got like an 8 angstrom structure. You can map them all back and do some analysis on local regions. Here's the structure you got. You can start seeing decent secondary, well, like 8 angstrom-ish structure. Okay, here's an in-house example of this user, not cited, wanted to look at the interaction between liposomes and some VLPs. And as you see, there's no fiducials here. We just aligned this with Tomo. And yeah, so they're interested only in these VLPs that are near these liposomes. So they need this 3D information. Tomography is the only way they can do this. Here's another example of a group who has interest in how actin deal with stress and strain, so pushing and pulling on them. And so they have protocols where they can stretch a single actin across a cryo-EM hole. So everything else here is just ice. There's some weird leopard skin ice in this particular scenario. With Tomo, you can align this tilt series. I don't think anything else can. So we have 3D information of a single actin across essentially buffer, a hole with buffer in it. Here is a tilt series. It looks more or less like noise, but there's actually HIV trimer in there. So this is if you want to understand how your single particle samples are behaving in your ice and how thick your ice is, then you can collect a tilt series. It may look like that if you have small things. These are actually not very small. I can do this with things that are about 10 times smaller. Here's an interesting object. So maybe this is giving you ideas for how to deal with your own project, or hey, I can actually do this with my project. Here's a strange stack of pseudo 3D arrays. Not sure what it is or if it's even biologically relevant, but we can do tomography on it, damn it. Okay, good. Here is an example of what you can do if you collect a tomogram of a single particle sample. So we had a user who, this is all published now, who had issues with making an initial model both for the single particle refinement and for fully picking the single particle images. So we collected a few tomograms, we got these structures right here, and we went back and we used these as templates for the single particle collection to pick all the particles. And then we used these also for what is a de novo initial model for the single particle refinement, which went down to 4 angstroms. So it was an essential component of this project. And that was cool. Okay, here's another project that recently got accepted to Nature here. So we had a reconstituted system of liposomes, and these are protocoherence, clustered protocoherence, which, just for context, in the brain at different, or in between neurons, we're trying to understand how individual neurons avoid themselves. And so we made a reconstituted system of these cadherins, which are known to be between each of these, whatever, axons, or I'm not a biologist, and and then studied the structure of them. And you can't do any single particle on this type of sample. It's actually hard to even do any subvolume alignments. But in the actual individual tilt or tomogram, you can get domain level resolution, where each domain here is about 13 or so kilodaltons. And you see each of these little bumps as we go through one of the best parts of this tomogram. Yeah, so we can get roughly 13 kilodalton domain resolution. Let me set this pixel size. You could probably do a bit better. And then this is with the neural network EM2 segmentation. Micah did that. So we pulled out the structure of the proteins and of the liposomes. Then you can actually sit down and fit in the crystal structure, and it fit in quite well, and they were able to look at potential long-range interactions of these cadherins with each other. So that was a fun little experiment that only Tomography Cryo-ET could do. Okay, here's another example where somebody had a virus and it had different types of spikes. I'm not completely sure about the biology of this, but you couldn't do single particle on this because they're slightly different size, and because they wanted to know the stoichiometry of these spikes. So they went through, did an overpicking of every single spike on every single virus, went through alignment and classification, then remapped everything back onto the original viruses to understand their stoichiometry. Okay. You might also have situations where you want to understand you want to be able to classify stuff in 3D in your in vivo environment. So these are extracted bacterial flagella motors from, I believe, tomograms of maybe some type of, it must be bacteria, I assume. And then, so in Dynamo you can do quite complex, what is it? PCA clustering classification in 3D, which is quite useful, and it takes a lot of time. EM Clarity is also very good at classification. They're able to pull out different conformations of this flagellum motor, particularly the ring structure down here. Right there. How much time do we even have?
10 minutes. Yeah.
Dang. Nice. Okay. So if you guys have questions, just keep yelling them out. Here's the highest resolution published structure. There is a Twitter published structure that is high resolution, but it's Twitter. So, all right, this is an HIV capsid from John Briggs' lab. Florian Schur did all the processing, and I talked to him about how did he get like everything down to that resolution. Pretty much you get 700,000 like subunits, and you average them together, you can get that resolution. And he threw the kitchen sink at it when it came to doing all the processing. He tried every package. You really just have to dive in on the command line, understand exactly where your data is at every single step, and push it through manually through all these packages. So he ended up using Tom, AB3 Dynamo, and a bunch of in-house scripts, which, as far as I know, are just sitting in-house in John Briggs' lab all the time. But that's just how it is, because they're so complicated. Right, Krios K2 Super Res Energy Filter. He used fiducials. Vim Hagen collected it at Embl. Quite a wide defocus range here. He only used strip-based CTF correction, at least in the original paper, about 750,000 asymmetric units. And then EM Clarity, or Nova CTF, that 3D reconstruction CTF software, pushed it to 3.4 angstroms. EM Clarity got it to 3.2. I think Warp also got roughly the same, though these are Twitter unpublished claims. Okay, here is, I don't know how useful this is. Okay, you zoom in, you get side chains. Okay, good.
Yeah, we went over this.
You went over this.
This was the journal club.
Okay.
Last Wednesday.
Let's get out there. Here is an old, but somewhat useful list, and maybe it's better for you to sit down in your PDF and just stare at this for a minute. And there's some better structures out there now. Different structures published, and what they use for tilt series alignment. Almost everybody uses fiducials. And then what defocus they're at, how they CTF corrected it, etc., etc. Pay attention to the number of asymmetric units, though. Usually, I guess, between 10,000 and several 100,000 will maybe, as long as your actual structures permit, get you below a nanometer resolution. Okay. Yeah, okay, so we already talked about, for limitations, the sample thickness, camera, specimen damage. You're also always limited by your pixel size. You have isotropic motion, or anisotropic motion, I wrote it like that. You may have inherent flexibility, of course. Now, two things that I haven't talked about here are when you're collecting, every electron that's hitting your sample is changing it in some way. It's making it move, and it's moving in 3 dimensions, definitely, because you're tilting your sample, so it's going to keep moving in different directions for every single tilt image you're collecting, and your ice is going to be bowing. So what I mean here is, here's a little video, again from Journal of Twitter, where first image is 0 electrons, and the second image is after 20 electrons, and the exposure area was right over here. All these little black dots are gold beads, and this thing is whatever he was collecting as Tomhagen. But you can see that much change in the overall structure of the area that you're collecting. That is, what, about 10 nanometer change based on the gold bead size, just by looking at it. And so that's not just 2D motion, or I mean XY motion, it's also moving in Z for sure, because I think this is a tomography collection. And so you have your objects are at, let's say, the first image. They're in some orientation or some configuration like this. Then after some number of electrons, like something will move, this may move over here, this may move down here, this may move up here, etc. Everything's moving in a 3D anisotropic way. And also your ice in that hole is often just bowing like this. So all these movements and probably others are combining and different charging effects to get different motion of your objects in the image. Now if you have gold beads, you can use some software called Tomo Align, which is the same guy who wrote Tomo CTF and other pieces of software, where it assumes that your gold beads are in a flexible environment on a per tilt image basis, and it will try to unflex, unwarp every single tilt image based on this assumed and discovered global 3D movement. And it will try to draw a vector field based on these gold beads across every single tilt image, and I think in 3D space too, and reconstruct based on that. So that'll get you potentially a little bit of resolution increase if you're using gold beads. If you're not using gold beads, and this might be better even if you are using gold beads, is to jump into a piece of software like EM Clarity or maybe Warp, where it will take every single sub-tilt series of every single particle and do a local refinement, like a single particle refinement on all those individual particles. So you get little tilt series that are just locally refined for every particle, and it does local CTF estimation and correction. And so the idea then, I don't know if anything does this, I think Warp may do this in the future. You take all your objects, like objects, you do sub-tilt series alignment on them, and so then you have the local motions of these things, but you don't know let's say this is in a cell, so you have maybe nuclear pores where you can't do this on, but you want to refine those also, so then you can interpolate back onto the original tilt images a 3D vector field based on these local motions, and then that will allow you to refine the unique objects inside of your tomograph. So that's like a dream right now. Something I have no time to get into, but if you're doing, I think we talked a little bit about Cryo-Clem, but 3D Cryo-Clem might allow you to get inside directed location of objects, either in FibSem or perhaps just directly in tomography, if you can go from the 3D Cryo-Clem to your Cryos or whatever and collect. All right, another thing I have very little time to talk about is rapid tilting. So currently it takes 15 to 30 or so minutes to collect a tilt series. And the main reason that it takes so long, Grant Jensen has realized, this is recent work by his group, is that when you collect an image, you say, okay, camera, I'm collecting a new image. And it says, okay, collect that image. And then it says, okay, now I'm going to process that image and then shove it off onto another computer while you're tilting your stage. That starting collection and ending collection from the camera side is seemingly a bottleneck, because you can actually tilt the stage pretty quickly, and that quickness is exemplified here in this experimental data, where you can just spend down to 12 seconds or so, depending on your mag, to actually tilt the stage and collect. So what Grant did is say, okay, I'm just going to open my camera and then turn and just keep my camera open. I'm going to blank it, but I'm not going to actually tell it to process those images, those frames. And so you end up with a giant stack of frames of like 0 degree and then black frames when you're tilting, and then 2 degree and then black frames and 4 degree. And then you have to do post-processing on that stack. But essentially you can cut down tilt series collection time by an order of magnitude by using either a fast incremental or a continuous collection. Okay, now I've added this, and you can look at it in your PDF. Misha Kudryshev and I sat down over in Frankfurt last summer, and we decided, let's make a giant workflow of all the possible things you can do in tomography. And here's how disastrously messy it is right now in the field. This is all just different processing steps and different software. So I'll leave that for you to digest in your PDF. Otherwise, I'm done. Questions? Yeah. Tomography is complicated. But it's fun.
Mm-hmm.
Always.
Yeah. I mean, single particle is just pretty much a couple buttons, so you should switch over to tomography today.
Yeah, it's too easy.
Join the club.
It's
Yeah.
dying science.
All right, if there are no questions, Wednesday will be our first tomography practical. So Alex will hopefully be here, since we'll probably be looking at a lot of the data that he has sort of accumulated for his earlier papers.
Yeah. I'll send out the worksheet so you see Alex how we take the class, and then it'll be more of an introduction, like how to play around with it and understand the software, because the second tomography practical with Mike will be more like they'll be more actually using it and seeing more install stuff and doing some more processing with it. So yeah. So I'll send out a worksheet for you guys, and you can install, and there will be problems installing the software. Come along Wednesday, and then we'll play around with tomography and IMOD and some of the nice tomograms and access collector for single particles.
So this will actually be hands-on. Download some software. It's pretty easy. IMOD installs into most things pretty simply.
Yeah.
It's just IMOD.
Yeah.
What we need.
So we'll send out a link that will send you to a webpage with instructions on how to download it. You can let us know if you have any issues. And then we'll probably send out a link to download some test data set that we'll start walking around with on Wednesday. And on Monday there's no class because it's President's Day. And then another practical after that.
On tomorrow.
That's tomography part 2.